everyone. It's 2025 and we've come together again. And I have to say, we are a group that works together. And to me, what that means is we both help each other. So remember, we have our speech study and we're only asking for 15 minutes once a month from anyone who might have ALS. You might probably have ALS, or you could be healthy, and we could use that voice also. There'll be a link in the chat, and please do us all a favor and remember how important each of us is on this journey. So welcome to another expert talk, and I will turn this over to our executive director, Christian Rubio. Welcome, Christian.
Thanks, McFinn, and thanks everybody for joining us this afternoon if you're on the West Coast, this evening, anywhere east of us in the United States, and thanks for everybody joining internationally as well. It is a time of change, of course, as we see in the United States, and it's a time of great kind of adventure, really, of understanding what comes next for all of us, and we're all in it together. We're glad to be here together as we gather every couple of weeks for another expert talk. Tonight, we have a really special guest expert speaker, Dr. Molly Gale Hammell. Dr. Hammell is Associate Professor at the Institute for Systems Genetics at NYU Langone Medical Center. She'll be talking about the three molecular subtypes of ALS, stress, inflammation, and TDP-43 pathology. And a little background on Dr. Hammell is that her lab combines techniques from computational genomics and molecular neuroscience to better understand the changes that give rise to neurodegenerative diseases. The lab has a special focus on ALS and frontotemporal dementia, FTD. And in particular, the group are interested in whether a viral-like sequence in our genome called transposable elements, or TEs, are contributing to ALS and FTD. If you're interested in collaborating or joining their lab, you can get in touch with them on their website at mghlab.org. And they are generally interested in using iPSCs to model human disease, building better algorithms for large-scale multi-omics profiling, and working together with consortia of different types to collaborate on neurodegenerative disease research. Some of the specialties that the lab and Dr. Hammell have in-house are gene expression data analysis, next-generation sequencing analysis, bioinformatics, computational biology and statistics, and molecular biology, genetics, and developmental biology, transposon small RNAs, and RNA biology. It's a great pleasure to welcome Dr. Hammell to speak to the community, and as always, you can ask your questions anytime in the chats. We will save the Q&A for the end of the presentation, but by all means, as you go along, if something jumps out of interest to you, please don't hesitate to ask a question then. And we'll moderate accordingly at the end to make sure your questions get answered. And I'd like to call Dr. Hammell forward now. Thank you.
Thanks so much for that really generous introduction. I'm really happy to be with the group today. So I'm just going to share some slides that I prepared for the talk today. There we go. And hopefully you guys can all see my slides. So wonderful. So thank you so much for inviting me to spend the evening with you. It's evening here in New York where I am. As I said, my lab works together trying to understand the basic biology of why ALS happens, and then combining that with looking at large-scale patient profiling data to try and see if we can match those up and say, we think this is what's happening, and when we look in the actual patient data, we have evidence that exactly that particular process is what's going wrong. So that's generally what we're looking at, and I'm going to tell you some of the results from my lab's recent work in this area. So there we go. So I have to tell you before I start, I am on the scientific advisory board of a company. It's called Transposon Therapeutics. It's a little unrelated to the work tonight, but that is an important disclosure. So before I get started in everything, I just wanted to actually tell you what these transposons are, just to give you a bit of a background. So these are sequences that exist within our genome. They are transcribed they exist in the DNA of our genome. They're transcribed from DNA into RNA, just like any gene would be. They code for their own proteins, and what those proteins do is they make a copy of that little transposon sequence and they insert it somewhere else in the genome. So this is exactly what viruses do when retroviruses come into our genome and try and make a new copy of themselves and insert that in so that they can start replicating and using our cells to make more virus, except this happens to happen in the human genome. So these were originally discovered by Barbara McClintock at Cold Spring Harbor, which is where I started my lab many years ago. She discovered these, believe it or not, in plants, but they exist everywhere. So transposons are these funny little sequences, but they actually are relevant to human biology. So the first human disease that was linked to transposon activity was discovered in the 1980s by a scientist named Haig Kazazian. So he was looking at trying to explain why patients in his study had developed hemophilia with no known mutation that they had inherited from their parents. And it turns out that a transposon had hopped from one place in the genome into the factor VIII gene that's required for blood clotting, and it broke that gene when it did, and this led to hemophilia in the patients. So this is the first one that was discovered, right? We now know of about a few others where these transposon-driven mutations have interrupted gene function and led to human disease. Right. So just to give you an idea of this scale, the protein coding genes, the ones that code for blue eyes and brown hair, all of those, that's about 2% of our genome. This was a big surprise when we finally sequenced the human genome about 20 years ago. And we were trying to figure out for a long time, what is all that stuff in the rest of the genome? And it turns out that transposons are about half of it. About 45% of your genome is these transposon sequences, these remnants that can hop around if they're given access to do so. So we think that this hopping around, this creating of new mutations, it's not super common. We think maybe 1 out of every 3 cells in your body has a mutation linked to a transposon that's hopped around and created a new mutation. And most of the time we're fine. So an endogenous retrovirus, I know you heard from Avinath at one of these. He studies endogenous retroviruses. And so endogenous retroviruses are one type of transposon, right? These are examples where a retrovirus has inserted itself into the germ cells when they infected a human host. Those germ cells meant that their children inherited that copy of retrovirus that inserted itself into those cells. And then all of their children will now have a copy of that virus in the DNA of every cell in the body. So this is one of the ways that these things can get into our genomes. Now, when they go in, they're broken. So they can't reinfect you or be caught by someone. For better or worse, they're stuck in your genome. And that's a good thing. Otherwise, we'd be passing these back and forth all the time. So even when these transposons, these viral-like sequences, aren't causing mutations, they can still be trouble by other means. And one of the ways in which transposons can cause trouble is that as they're trying to hop around, they'll create viral-like byproducts, cDNA, double-stranded RNA, proteins your immune system hasn't seen before, things that can be a source or a stimulus for the inflammatory innate immune system, and that we think might be a driver for neuroinflammation. So I'm just trying to give you a sense of what these transposons can do. So one of the things they can do is cause mutations when they hop around, and one of the things that they can do is look viral-like enough that they might actually alert our immune system, tricking a cell into thinking it's been infected when really it's just aberrant activity of these little transposons. So just to give you a sense of whether this is theoretical or whether this is really happening, I'm going to give you some recent articles that have come out about actual transposon activity in human cells driving inflammatory pathway activation. This is from my colleague John Sidivi's lab. There's another one where double-stranded RNA derived from transposons, they think is a driver for neuroinflammation in Alzheimer's disease. This is my colleague and friend Bess Frost's lab, right? And then, of course, you heard about transposon activity from my colleague Avinath at the last of these EverythingALS series, right? Okay, so that's pretty much the rest of the primer background. Things are sounding kind of grim though, right? I've told you that your genome is made up of more transposon sequences than it is normal genes. I've told you that transposons can hop around making copies of themselves, and even if they're not making mutations, they can otherwise damage the host cell in which they're active. And so one question you might have for me about this is, if these are so important and so dangerous, why don't we hear about them all the time? And why are we mostly okay? Why aren't they? In other words, why isn't every cell a Wild West of active transposons causing damage, right? And so the simplest answer to this is that most of the transposon sequences that exist in our genome are very, very old broken copies. They can no longer hop around for sure. And much of the other things that I've told you about, including encoding for proteins, they might not be able to do that either. So out of the millions of transposon copies in your genome, probably maybe 100 of them are actually dangerous. But that's still about 100 that we need to watch out for, right? Okay. And so what do I mean by mostly okay? Well, I've told you that, there was at least one human disease, hemophilia, caused by a transposon mutation. We think that in every tumor cell, there's probably 10 to 100 mutations that are induced by transposons. And again, like 1 in every 2 to 3 cells in our body probably has a mutation due to these things jumping around somatically after we're born. Okay, so I spend a long time studying transposon biology in my lab. And we had some hints that some of these processes might be at play in ALS. But I really wanted to know if that was true, if transposons have any relationship to ALS biology, and I'll tell you about that in a second. What might they be doing? Could they be causing new mutations? Could they be drivers in inflammation in this? Those were the big questions that we were trying to ask. And so why did the hunch that we had for why transposons might be involved in ALS is because one of the major proteins that normally keeps transposons silent is a protein called TDP43. And that's exactly the one that aggregates in ALS patient tissues in upper and lower motor neurons of the motor system, and the one that's driving ALS progression. And so we think when TDP43's normal function is lost, it's also losing this normal function in controlling and silencing transposable elements. So that's why we got here and why we think they might actually be related to ALS disease. So just really, really quickly, a little bit more background on TDP43. This is normally a protein that exists in the nucleus of our cells where it's controlling how other genes are turned on and off, right? So it's controlling splicing, export, small RNA biogenesis, all these things that are just about how a gene is turned on or off in a particular cell. And my lab is the one that showed that they're actually also controlling transposons, meaning normally keeping these silent and keeping them off so that they don't cause trouble. So we know that in the very early stages, TDP43 gets mislocalized out of the nucleus, so it's no longer performing those normal roles, meaning it's a loss of its normal function. And then in addition, it starts aggregating out in the cytoplasm, and those aggregates themselves are toxic. So when TDP43 aggregates are present, we think you've got two problems. So problem one is that the aggregates themselves are toxic. And problem two is that TDP43 isn't doing its normal job in controlling how other genes are used and also in silencing these transposable elements. Okay, and so this is just a slightly less cartoonish version. This is what those aggregates actually look like when we're staining for TDP43 protein. And you can see that instead of being in the nucleus in the center of the cell, it's out there aggregating and completely filling up the cytoplasm of these triangle-shaped neurons, these pyramidal neurons. And those are the ones that exist in the upper motor neurons in the motor cortex and the lower motor neurons in the spinal cord that are controlling movement, right? So we had all of this data that TDP43 controls transposons. We had all this data that TDP43 is dysfunctional in ALS patient tissues. And so the next thing that we wanted to do was to ask whether or not this biology of TDP43 controlling transposons and other viral-like elements was actually clinically relevant. And so to ask that question, I teamed up with a very large international ALS consortium run out of the New York Genome Center. So my colleague Hamali Fatnani was the one who was coordinating that consortium to look at lots and lots and lots of ALS patient tissues to try and get some answers about what exactly was going wrong in the motor cortex and spinal cord tissues of affected patients. And so there were a lot of clinical centers as well as research centers who were contributing to this. Target ALS was a big backer of the Genome Center Consortium. And of course, there were hundreds of ALS patients who donated tissue, clinical records, time and effort to help with this study. And so our plan of attack was that we were going to sequence the DNA, the genomes, to look for mutations and other things that were going wrong in those particular tissues. We were going to look at the RNA to know what genes and transposons are active. That tells us which genes are being used and whether transposon activity is elevated in those patient tissues. And finally, we're going to stain for the proteins like TDP43 that'll tell us whether or not that pathology is present in the particular piece of tissue that we're looking at. And in this case, motor cortex and spinal cord. Right? And so the first answer is that we definitely saw elevated transposon activity in motor cortex tissues, and those were exactly the same cortex tissues that had dense TDP43 pathology. So the more cells that looked like this, where TDP43 was completely filling the cytoplasm and no longer performing its nuclear roles, the more we saw that transposon activity when we looked at the RNA expression in those patient samples. But one of the things that you might notice is that this is a small set of all of the patient samples that we were looking at, and not all of them had this particular signal. And that was a little bit surprising to us because we knew that all patients should have TDP43 pathology somewhere. So what was going on with these other patient samples? We had type 2 and type 3 here, where different genes were being misexpressed. So this is all ALS patients, and all of them look different from the control samples that we had, right? But they look different from the controls in different ways. So the first group we looked at, they absolutely had that transposon signature and that TDP43 pathology signature. But what was going on in the other two groups? So my lab used some of our machine learning methods to try and figure that out. And the first thing that we figured out is that the largest group of patients, about half of the ones that we looked at, had markers of things like oxidative stress and ER stress, autophagy markers. So these are all things about mitochondrial dysfunction, right? So it looked like the pathways that normally power cell function were really stressed and not performing well in these particular samples. And that was different in the sense that they didn't also show TDP43 pathology, and they didn't also show that transposon activity, right? And there was a final group. This was the smallest. It was about 20% of all of the patients that we looked at. And what was really going on in these patient samples is rampant and systemic neuroinflammation. So microglia are the inflammatory cells of the central nervous system. And these cells that are normally sort of small and rounded up and sensing whether the neurons around them are okay, were instead hypertrophic. They were overactive, and they were driving these inflammatory pathways. And we saw these all throughout the tissue, rather than where in normal cells or ones that don't have this inflammatory activity, the microglia are relatively silent, right? And mostly in their homeostatic roles. Okay. So this was the summary of that study. We definitely had transposon activity and TDP43 pathology. Those things go together, and they were always correlated in a subset of our samples. But along the way towards trying to understand whether transposons were related to ALS biology, we also happened to find these two other subtypes that we were surprised to learn were distinct, right? So there was a distinct set for which neuronal stress pathways were the majority of the molecular processes that were going wrong in that particular set of patient tissues. And then there was this third type where neuroinflammation, so rampant glial-mediated inflammatory pathway activity, was what was particularly going wrong in this set of patients. Okay. So this was published a couple of years ago. We were really excited about this, but it opened so many new questions, right? So you can think to yourself, well, there were only 200 patient samples in our first pilot set of studies. What if we keep sequencing from new clinical centers? Are we just going to keep discovering new subtypes, right? When we have larger studies and more power to look through them. So that's one of the questions we had. And then another question is, what exactly is a subtype, right? Are these really three independent and completely distinct forms of ALS disease? Or could you imagine that maybe there's a stage where first the cells accumulate stress, eventually they'll accumulate TDP43 pathology, and then perhaps inflammatory pathways get upregulated at the end as a sort of cleanup crew, right? That's one particular model you might have to explain why there would be three distinct pathways for what should be one disease, right? So I'll answer the first question first, which is that we now have, with the latest distribution in a preprint that my lab just published, 700 samples from both spinal cord and motor cortex, and we don't actually see any new subtypes arising. We just get the same three over and over again, right? So it's either that pathology-driven transposon subset, or it's the inflammatory subset, or it's that, neuronal stress subset. Those are the three that we get. And I could go on and on if you want to ask me about the data of how we decided that, but I'll just leave it there for now. Finally, there are actually clinical correlates of these molecular subtypes. So these molecular subtypes aren't only something that we see when we look at the RNA data. If we actually try and go into the records to see how long the disease lasted for the patients that had one subtype or a different subtype, we actually do start to see that, for example, TDP-43 pathology and associated expression of these transposons, that was the strongest, most correlated thing with disease duration, meaning the more pathology you have, the more transposon activity you have, the shorter the disease duration was for that subset of patients. So this is 50 months or about 4 years right here. And most of the ones with the strongest signal, the disease duration was really short. And that was different from where we were looking in the spinal cord, where in the spinal cord, it was neuroinflammation that was really driving those shorter times from when a patient is diagnosed to when they either passed away or had a tracheostomy, right? So there are clinical correlates that tell us that these pathways that we're finding in the patient tissues are telling us about things that are relevant to how this disease progresses, right? So how might this help us fight ALS, right? That's the ultimate question, is this actually going to be useful going forward to know this information about whether somebody's in one subtype or a different one? And we're working on that. We're working right now, especially on trying to look for biomarkers. So everything I told you was discovered either in motor cortex or spinal cord. These are tissues we really don't have access to when the patients are diagnosed. And what would be useful is to know this information when a patient is diagnosed, when maybe we can do something about it, right? And so we're looking to see if there's any kind of biomarker that we can find in plasma or whole blood or CSF or maybe even muscle biopsies that will tell us which particular type of ALS one of those patients had. And in addition, even if it's a stage, maybe we could know what is the particular molecular process that's going awry and really driving disease progression at that time. Right? So in either case, what we really need are biomarkers, right? Because you can imagine that you might be able to stratify patients by whether or not the main problem that's going on, for example, is TDP-43 pathology and transposon activity, which we can actually potentially target with some antiviral compounds. That's in some other preclinical studies that I don't have time to tell you about. But you could also imagine that, for example, some of the anti-inflammatory compounds that are being tested might be more likely to work for the patients where, that ALS glia pattern of inflammatory processes is actually taking place in the patient samples, right? And finally, maybe for some of the patients who are just really experiencing these neuronal stress pathways, these mitochondrial dysfunction pathways, that there'd be other things targeted for them. So at this point, I've talked for too long already, so I'm going to stop. And I'm just going to thank my lab, the students and postdocs who really do this work, the consortia that I'm a part of, and all of the other researchers who've been taking part in that and trying to help get some answers here, the funding that allowed us to do this work, and of course, all of the patients living with ALS who've been donating their time, samples, and energy in this fight. And thank you.
Thank you. Yeah, thank you so much, Dr. Hammell. This is great introduction to, I think, what's also going to lead to a robust conversation as well. We have gotten a few questions already, and I think to your point, it is great to have an extended conversation now, where we had a presentation recently about HERK, and now to continue to talk about the impact of these kinds of virus-like or infection-like impacts in the disease. So. My first question that I've seen in the chat, and Natalia and I will alternate, would be: do you have a research partner in Australia, or where else is the research being done in the consortium?
Yeah, so most of the research partners are either here in the US or in the UK. So like UCL is one of them, right? There's partners in Edinburgh, there's partners at the Charity Hospital in Germany. So there's a bunch of different partners who've joined the consortium. I think it's exciting to think that we all might be able to work together. So everyone who joins the consortium gets access to all of the data, right? So anyone who's interested in that can really apply to join the consortium, and they'll get access to everything, including all of the software that's used to find these things.
Thank you. The next
No collaborators in Australia. I didn't answer that one specifically. Sorry.
Thank you.
Yes, our next question is a bit more about methodology. So the question is CSF a better source fluid than serum for creating iPSCs for this study?
Oh, for creating iPSCs, that's an interesting question. So everybody has a different idea about this, right? Some people think that fibroblasts that are directly transformed into neurons will do a better job because they'll carry some of the epigenetic marks that they had in the original tissue. And so we're really just kind of trying all of the different ways to model them in hopes and then comparing them back to the patient tissues to say which of these looks most like the patient samples that we have, and then say, okay, that's the one that we're going to move forward and try to test things in.
Thank you. And the next question is, do any individuals have any more than one subtype from the way you've done the subtyping in the study?
So that's a really interesting question. So we happen to, for about 200 patients, we happen to have matched cortex and spinal cord from the same patient, and it doesn't always line up, right? So one of the things that we discovered, and this is also published, it's available on a preprint in PubLed or on my lab's website if you want to really dig into the data. But one of the things that we figured out is that inflammatory pathways are really common in the spinal cord and much less so in the motor cortex. And we don't really understand the reason for that. We don't understand if that's because inflammatory pathway activation happens more quickly there, or if those patients have a greater burden of disease in the lower motor neurons of the spinal cord. But we're hot on the trail of that to ask it. What I can tell you is that separately, in pathology in the cortex and inflammatory pathways in the spinal cord, those are the two things most correlated with really short disease duration, right? And so we think that is something about how it presents in the two different tissues and something about what's driving it.
Thank you.
That's wonderful. Thank you for your detailed answers. The next question is more about the subtypes, which you mentioned. And we have a question asking if TE subtype, I think it was the second one, is separate from HERV-K positive type, or maybe you can talk a bit more broader about different subtypes and how it's related to HERV-K.
Totally. So HERV-K is really interesting. It's one of a number of different kinds of transposons. And for the most part, they all go up together, meaning that if you see one kind of transposon, you're almost certainly going to see HERV-K as well. But that wasn't always true. And occasionally we would see HERV-K activity when we didn't see the whole rest of the transposon types. And we're interested in that because we want to know why that would happen, right? And so we're looking to see whether or not, in addition to TDP-43 pathology, there's anything else that could drive individual types of transposons such as HERV-K, right?
Thank you. Next question is, are bulbar or limb onset ALS associated with a particular type of subtype in the way that you've been characterizing them?
Yeah, so I can tell you that bulbar ALS was the most likely to have inflammatory pathways active in the spinal cord, right? That was absolutely true. Limb onset, there's my lab has been trying to answer the question of whether or not it started in upper motor neurons or started in lower motor neurons. And that's a little bit separate from the question of what's limb onset, because you can have both, right? But we're actually trying to figure out whether or not one of the two of those is going to be more correlated with whether you're in one of those subtypes or a different one.
So the next question was about the details on TDP-43 pathology in ALS OX and ALS glia group, and if it's like present there or at all, or how it's different.
Yeah, maybe I will reshare one slide. I prepared this one just in case I got asked a particular question. One of the ways that, let's see if I can get it to advance. One of the ways that we're thinking about this, so if you think that maybe a subtype is a stage, right, then you're imagining that over time, patients are sort of going through these different stages where first mitochondrial dysfunction and ER stress is really driving what's going on, and that's particularly a first stage, right? And then later, all of that stress is eventually driving accumulation of aggregate pathology, right? And after some time at which that aggregate pathology has been accumulating in cells and really damaging the cells in which it's accumulating, perhaps inflammatory pathways are getting upregulated afterwards as a sort of last thing in a cleanup crew. And the data that we have is somewhat mostly consistent with this model in the sense that when we see mitochondrial dysfunction and ER stress pathways mostly active, we're almost certainly not seeing inflammatory pathways active in those same patients, and we're seeing very little pathology, right? Whereas for the patients who have inflammatory pathways active in cortex, they also have a little bit of pathology, and we don't know if they have, slightly less pathology than here because some of the neurons have been lost, or whether or not there's just something else that's driving inflammation in these particular patients. So this is the framework that we're working with and trying to think about how these subtypes relate to each other and how they might relate to pathology and accumulation of inflammatory pathway activation. But like I said, this is a model, and we haven't conclusively proved that this is the way it goes. But this is the way we're thinking about it.
Thanks, Dr. Hammell. Next question up is there any correlation between disease subtypes, as we've been discussing, and familial versus sporadic disease?
Yes. Yes, the answer is yes. Generally, we don't have enough familial patients in our studies to do absolute case control calculations, but I can tell you a few things. So the SOD1 patients and the FUS patients, they never develop TDP-43 pathology, and they almost exclusively grow into that ALS-OX neuronal stress group. That's where they're going. Right. The C9orf72 patients, that's the largest mutation set of patients with mutations in familial ALS. They're sort of split. They get a lot of TDP-43 pathology, and a significant number of those patients also get a lot of inflammatory pathway activity. And so we'll see the patients be either in one of those two groups when it's C9orf72 patients. The less common mutations, right? So the optoneurons and the TBK and the NEC1, these we just don't have enough patients to actually say anything one way or the other about whether or not they're splitting into different groups. But we're certainly trying to model these in iPSCs to say, if I model a particular neuronal cell line to carry one of those mutations, are those pathways that are dysregulated in those cells going to look more like one of these subtypes or another one? And that's how we're going to try and answer that question.
Next question we had was related to COVID vaccines, and if there is any relation between that and inflammatory pathways in ALS.
So that is a really great question, one that we have not looked at all. So I would have to speculate, right, if I were going to say anything about it. We don't have any data on ALS patients who've either had COVID vaccines or had COVID. But it's interesting to, I think, speculate about whether or not there might be interaction between different kinds of retrovirus infections or immune activation.
Thank you. All right. Here's kind of a multi-part one for you. Since transposons are found in so many other diseases, what happens when you do similar analysis on other neurodegenerative conditions? Have there been those studies done that you can speak to? And do you find similar clusters? And are the clusters clean and separate in other diseases like you're seeing here?
So that's a wonderful question. I am part of a team of collaborators that are working to see whether or not transposons could be triggers of inflammation in Parkinson's disease, in particular because we do know that inflammation spreads alongside alpha-synuclein pathology in that particular disease. That said, I do want to back up and say that not all transposons are always expressed together, right? So each transposon has its own promoter that drives its expression in different cell types. And so I might expect that while transposons generally might be involved in one disease or another disease, it might actually be a different one that is upregulated in Alzheimer's, for example, where they seem to see some transposon activity, or Parkinson's disease in the dopaminergic neurons that are most important to that particular disease, and where we think we might actually see some signal for transposons being accompanied by inflammation in that setting, right? So I can tell you that in ALS, which is the one we've looked at most, it really is HERV-K and line 1 and a subset of ALUs that we're seeing there. And I think the question is open of whether we see exactly the same transposons, exactly the same transposon activity in other diseases when it's not linked to TDP-43 pathology. And that's one we're trying to go after.
Thank you. The next question, if you're still looking for more samples, our community is interested, and if you're, I guess, continuing running this study.
Yeah, so the study, we're continuing to run the study for sure, right? I think they're definitely looking the ALS registry, Target ALS and the New York Genome Center Consortium. They're always looking for people willing to contribute, and I think everybody appreciates the fact that there are patients living with disease who are willing to contribute. That's an amazing donation to science.
Thank you. Next question, are there any biomarkers, labs, or tests that can help stratify along the same lines that you're doing here in terms of subtyping?
So that's exactly what we're trying to figure out right now. So we have we have a subset of samples for which we had blood and CSF for those patients, and then we also, when those patients unfortunately passed away, they did donate brain tissue and spinal cord so that we can look and we know exactly what kind of ALS they have, and then we look back and say, can we have predicted that, right? And so we're trying to go after that right now. I'm not positive it's going to be exactly the same genes expressed in the two tissues, but since we have the matching, it doesn't have to be. It could be that a different gene is or marker is expressed in CSF when these pathways are at play in the relevant tissues, and hopefully with the strategy we have, we'll still be able to catch that. I can tell you that a separate group replicated our study with an independent patient cohort, and they did have blood available for a large fraction of those patients. This was a study by Marriott et al. That was published a couple of years ago, and they were actually able to see subsetting of the patients into subtypes, and they had found exactly the same subtypes that we did in their samples. And so I think there is hope that between all of us trying to look for these biomarkers in every tissue that we can get access to, we might be able to hone in on a couple of things that overlap and that replicate between our studies.
Thank you.
I think it's partially related to this question, what you just answered, but to expand a bit more, is there any genes associated with each subtype, or if you've seen any correlations with known genes based on each subtype?
Totally. Yeah. So I'll just name some out there. So like the neurofilament proteins that we know respond to neuronal stress, those are definitely one of the markers of that ALS OX group for which stress pathways are the main driver of that particular subtype. And so that's exactly one of the things that we're looking for. We're looking with proteomics data in CSF and blood to see, when we see elevation of these neurofilament proteins, is that telling us that the patients happen to be in that OX stage or in that OX group? right? So that's one example. There are certain inflammatory markers that we're looking for, interleukins and other chemokines and cytokines that tend to be released by cells for which when inflammatory pathways are active. And so we're looking for exactly those same things there again, right? And I can tell you there are some studies that have seen those inflammatory biomarkers in patient samples. And I think the question is, if we had access to cortex from those same patient samples, would we see that those are the glia patients, the inflammatory patients, right? And then for the transposon subset, there's a number of things that we're looking for, including the fact that phosphorylated TDP-43 itself might even be released when the patients have accumulated a lot of TDP-43 pathology. And we're also interested in whether or not the transposons themselves can actually be detected outside of the central nervous system. So those are, like specific examples of all the things that we're trying to look for in those samples.
Thank you so much. I'm going to include the compliments here because you have received compliments all along the way. I haven't read some of the compliments just to keep the questions going, but you are getting a lot of congratulations, and it seems like excellent research. So one of the questions also is, in layman's terms, some companies are claiming they have a sporadic ALS cure that stops TDP-43 aggregation. Are you saying that one medication for a sporadic ALS patient might not work for another sporadic ALS patient, that we may need 3 cures or cocktail, or how would you characterize what would likely be needed based on this subtyping?
Yeah, so I think it depends on whether you think subtype is stage or whether you think subtype is independent disease type. And that's exactly the question that we're trying to answer so that we can answer the question you've just asked me, right? If you were able to block TDP-43 pathology, and if you at least stop it from spreading, that would be fantastic. I would love to see anyone who can show that that's true, and I would be very excited for it, right? If you imagine that desilent activation of transposons and inflammatory pathways, that all of that is downstream of TDP-43 dysfunction, and we were able to block TDP-43 dysfunction, then maybe that would be enough to take care of those two, right? And the question would then just be, can we prevent them from ever getting to the point where TDP-43 dysfunction occurs, right? So that's one possibility. If these really are 3 different patients, right, and inflammatory pathways are driven by something else that's unrelated to TDP-43 pathology, then maybe you would have to think about something else. But I think regardless of whether any of those things are true, patients will present sometimes with inflammatory pathway activation very early in disease, right? And I think we need to be able to have something that will catch the molecular processes that are occurring in that patient at that time, regardless of whether or not that works for someone else, right?
Yeah.
We have quite specific questions, but I think our participants are interested to see if they can know more which subtypes they might have. So do you know if blood samples from participants in Healy platform trials at Forbes Norris in San Francisco were included in the patient data set, which led to the discovery of 3 subtypes? And if so, can it be allowed, or was it allowed to share with the study participants?
So, okay, so I'm going to take 3 different parts of it, but the first part I'm going to take. So all of my data sets get released to the public, meaning like anyone who joins the consortium and agrees to abide by the rules can have access to any of that data. The patient data is all anonymized, right? So in order to encourage people to submit samples and to make sure that they feel safe releasing, that information, there's no way I have access to the names or identities of anybody who is in the patients. And that's a good thing, right? Like, we want this to be about finding things that are about ALS that would help us predict how to help individual patients later. And so I'm kind of going through the questions backwards, but then the very first one was, are any of the Healy platform patient samples in the consortium data set? And the answer to that is no, but only because the consortium data, the first paper that we published was in 2019. And so the patients had to have passed away prior to then. And that was before the Healy platform trials had started. right? But the Healy platform trials do include collection of material from patients to see who is and isn't responding to some of the drugs that are being trialed there. And I think that's going to be really interesting data to dig through to see if we separate the patients into ones who did actually have some slowdown of disease progression while on an anti-inflammatory, for example, or one of the other targeted therapies, could we see that there's something different that's being expressed in the CSF or blood from those patients than from other ones? And I think that's a really interesting analysis that could be done to try and get at the question that you're trying to ask, right?
Perfect. Thank you. Thank you so much for the explanation. Another question here. Were the subtypes derived from transcriptomics data of motor cortex and spinal cords of ALS patients? And do other types of data, e.g. proteomics, show similar subtypes upon clustering?
Yeah. So so those are really wonderful questions. They were originally identified in motor cortex because that's the subset of samples for which we had the largest number of patients when the study was originally done. So we now redid the study, and we've just released an update to that this year. So the preprint just got posted a few months ago. We now have 719 patients, and we have thousands of samples from them. It's still transcriptome because it's the easiest and fastest way to get lots and lots of samples from lots and lots of patients, right? In that case, we redid the discovery to make sure that there's nothing that we're missing. Like if we separately look in spinal cord, or we separately look in motor cortex, or we pool them all together, is there anything new that we would discover now that we have this very large set of patients? And the answer to that is no. We got the same subtypes in spinal cord, except that it was really just inflammation or stress, and the same subtypes in motor cortex, right? We have been using immunofluorescence and immunohistochemistry to make sure that some of the genes that we were looking at are actually misexpressed at the protein level, and that validates, so that's definitely true. And now we're actually trying to get proteomics data from a subset of those samples, but it's much more expensive and takes a lot more time to get large-scale proteomics data going, so that one's just taking longer, but it's going to get there.
Thank you.
Thank you. Next question is about any correlation with recent finding around cryptic splicing events and potentially some of the subtypes which you identify, and specifically ALS-DE or others.
Yeah, absolutely. So, one of the things that we did to verify that the ALS-TE subtype really is about both TDP-43 pathology and transposon elevation was to go in and look for those cryptic splicing events to see if those were most correlated in the ALS-TE group, because they should be, because those are downstream of TDP-43 misfunction. And that was exactly the case, right? So the more TDP-43 dysfunction we saw, the more transposon elevation we saw, the more of those cryptic splicing events that we saw, including STAFMA2, UNC13, the sort of famous TDP-43 cryptic splicing events. And one of the things that we're hot on the trail of right now is whether or not they're actually accidentally splicing into some of the transposons that are interspersed in the introns of those genes. But that's a very technical detail for people who are interested in that piece of it.
Thank you. This question you partially answered before, but I'm going to revisit it a little bit just to hone in on it. Is there any correlation between neurofilament levels in the serum and the different subtypes? If you could just maybe revisit that a little bit more.
Yeah, so that's the one that's ongoing work, right? And is super preliminary. So, we have a panel when we're doing proteomics in serum and CSF. We only have this for a subset of patients for whom we have that data matched, meaning like we have either motor cortex and or spinal cord for that patient, and then we also have serum and or CSF for that patient. And we do see a subset of patients for whom they have elevated neurofilaments, and we're just trying to prove right now that that really is about whether or not they have that OX type, and not necessarily just about neuronal loss at the time, because that's one other expectation for what those neurofilaments might represent when they're seen to be elevated in serum or CSF. Right. And so hopefully that study will be finished very soon, and in the next couple of months, I'll be able to tell you exactly what the answer is for that one.
Thank you. We have this kind of comments, and the community wants your opinion on that. So, with the current inability to identify a biomarker to predict or accurately stage the disease process, it seems to make sense that from the therapeutic perspective, you should target all the stages simultaneously. And the question is, what do you think about it? And do you agree with that or not?
Yeah, I mean, I think that's an open question, right? There is a little bit of debate about whether or not certain kinds of inflammatory pathway activity might actually be slightly protective at certain stages, and whether or not inflammation is always bad or might at some point be protecting some of the processes from spreading. And I think that's the only worry that I have about sort of targeting all of the different potential subtypes at the same time in a patient who might really only have one or the other. Right. That said, when we have no better option, I think targeting whatever you think would be most useful might be a good strategy. And I'm going to leave that to the clinicians and the ones running the clinical trials to make those decisions. But I do think that by the time that most patients are presenting with serious dysfunction and are and losing neuronal function, TDP-43 pathology is very likely to be rampant and is a very good target.
Following some of the other questions, would primate injection studies of altered TDP-43 suggest that this is a secondary pathogenesis pathway after oxidative stress pathogenesis pathway?
So that's a question, right? Whether or not, ER and mitochondrial and oxidative stress always leads to pathology and then always leads to inflammation. We do. So, I said that was a compelling model, but there's some interesting things about it that make it hesitate to say that explains everything. So for example, for the SOD1 and FUS patients that never developed TDP-43 pathology, right? Obviously they're not going along the same trajectory that everyone else is. There's some indication that some patients develop inflammatory pathway activation early before accumulating a lot of TDP-43 pathology. Those might be certain genetic subsets, so perhaps that's just different from sporadic disease. I think what's underlying the question that you're asking, though, is whether or not some of the things that we're seeing are secondary downstream effects and not the important thing, right? That, for example, transposon activity, I always get asked this question, whether or not that's just everything is going wrong in a cell that's dying, right? And so in some animal models, so mouse models where they've built in, elevate TDP-43 aggregation and or transposon activation, targeting, and also in fly models, believe it or not, of TDP-43 pathology, targeting the transposons themselves and targeting TDP-43 pathology itself was able to stop progression of dysfunction and progression of degeneration in those diseases. And so I think that is early evidence that, even if these aren't the main early driver, they're at least contributing to the disease such that targeting these pathways is enough to slow the degeneration and slow the progression of dysfunction.
Thank you.
Thank you. Could you share a bit more thoughts on triggers for the ALS OX?
Yeah, that's a really interesting question, right? Whether or not, I think a lot of people have proposed what the triggers might be, right? Different ideas about what the triggers might be, whether or not so neurons are highly active, and depending on the particular metabolic pathway that they use to generate energy, some of those pathways are more likely to generate reactive oxygen species than others. So whether they're using glycolytic processes or other pathways for energy generation, they're more or less likely to induce reactive oxygen species and oxidative damage. And that's been one proposed mechanism for how mitochondrial dysfunction might arise in an aging population in neuronals that are metabolically stressed, for example, right? I don't know that that is the cause. That is all speculation, and everything that I'm answering with this particular question is speculation. But I am talking about, neuronal models or animal models where they've tried to understand what might actually be driving these neuronal stress pathways, right? There's certain other environmental toxins that can do it, right? And the question is, what is actually doing it in patient samples? And I think that's still an open question.
Thank you. When do you think you'd find out more whether the subtypes are truly 3 different diseases versus different stages?
Oh, I think the biomarkers are going to be a really good hint, right? Because if we can see that so for example, there's 2 different possibilities that we're going to get there, right? So either the biomarkers that they had at the time that they were diagnosed match exactly what they had when they passed away. And in that case, that is the disease they had, right? If, however, the biomarkers that they had early told you at which stage of progression they were at, and later when we actually got access to CNS samples, we see that something different is going on, then it's stage, right? And so what we're trying to do is we're trying to figure out, especially if longitudinal samples could be taken, right, whether or not that will tell us this is about stage, or whether or not it's always the same kinds of things that are being detected in markers in those peripheral tissues, and therefore it's really about a different disease.
There is a question to understand a bit more details about TDP-43 accumulation per se, as it drives neuroinflammation, but the idea is what type of drugs can actually stop the process of TDP-43 accumulation to actually not allow the neuroinflammation process even start, if you have any idea.
That's a holy grail. Like literally every ALS researcher would love to be able to stop or prevent TDP-43 pathology accumulation. There's some really interesting work from James Shorter's labs at UPenn. He's got these chaperone proteins, which are proteins that refold misfolded proteins and send back a functional version of them. So he's got some really interesting work in that area. I think there's other people who are interested in that as well. There's this idea that TDP-43 auto-regulates itself. So when it senses that there's not enough TDP-43 around, it shuts itself off. But if TDP-43 is mislocalized out in the cytoplasm, it can't go back into the nucleus to the genome where the gene is and shut itself down, right? And so there is this idea that there's a snowball effect going on, that as TDP-43 starts to mislocalize, as it accumulates out in the cytoplasm, it can't go back in and shut itself off. And that if we could just send, clear some of the aggregates so that we don't accumulate more and more of it, right, and send some functional TDP-43 back in, that we might be able to slow things down. And so there are some people who are interested in making TDP-43 sensors that can sense whether TDP-43 is functional or not and shut itself down when it's clear that aggregate pathology is present. Right. And so that's unpublished work from some other researchers that I think is really exciting, and that might be able to, at the very least, stop further accumulation, even if it can't, absolutely refold TDP-43 protein itself.
Thank you. I'm going to tie this next question back to the question that I asked you just shortly before about subtyping in the 3 different diseases versus different stages, and the time it takes to get there and the kind of collection you need to make to get there. How can AI help? And are you looking at any ways in which AI can be implemented to potentially accelerate or better delineate these findings that you're trying to get to?
Yeah. So, AI is a funny word. It means a whole bunch of different machine learning methods that you're using to try and figure out what are the likely drivers behind the patterns that you're seeing, and in some cases, de novo pattern detection. So my lab builds these large neural networks, convolutional neural networks, that we're using to try and accurately classify patients into one subtype or another. That's one version of AI. Right. And then I think the next question is, once you train a model to be able to detect which subtype it's seen, you can then go back in and ask the model what it learned. so that you can maybe find out some more interesting information about why it put a patient into one group or another, and then figure out if that's going to be useful in helping you understand what those subtypes really are. What is the most relevant information that allows you to distinguish subtypes from each other, and whether or not that might help you discover it in other tissues, right? So if you train it in spinal cord, you train it in motor cortex, can you run it on some of these other tissues and see how well it performs there, and whether or not there's something that it's learned that does a better job than just, standard case control differential analysis, right?
Thank you.
Can you please clarify a bit more about TDP-43 pathology in other subtypes, not the ALS-TE? It was clear that it's mislocalized in this subtype, but since majority of ALS patients have elevated the TDP-43, just wanted to understand a bit more the pathology across all of the subtypes.
Totally. Yeah. So, that's a great question. So we went back in and we actually stained to look for TDP-43 pathology. And that's one way to do it slowly. We can do that in about 100 patients, but we can't do it in all 1,000, right? So then the next thing that we did is we built cell line. We took advantage of a data set where they had sorted out neurons that either did or did not have TDP-43 pathology, and then deeply profiled those to see all of the things, all of the signals that happen when TDP-43 pathology is present in the transcriptome. And the reason that's useful is you can go back in and then score every single, single cell or every single transcriptome sample and ask how highly does this score for the signatures of TDP-43 pathology? And you can do it without having to do stains of tissue slides, right? And so
Watch the ALS presentation.
So that's the technique that we used to try and figure out how much TDP-43 pathology is present in each of the samples, regardless of which subtype it's in. And what we saw is that both the ALS-TE and the ALS-GLIA patients highly score for signatures of TDP-43 dysfunction, right? So the ALS-GLIA patients have it too. That's just not the majority of the signal that's going on there. Right. And we can't tell if that's just because it's hidden underneath the signals from all of the microglia and astrocytes, those inflammatory cells that are driving dysfunction there, or if because there's been some neuronal loss there. And so we use single cell data to figure that out, and we think it's a combination of both. So that's the first part. And then the second part, and I realize I'm getting super detailed and super technical here, but I'm going to say it anyway. Every ALS patient, except this odd one of us, has TDP-43 pathology somewhere, but they don't all have the same degree of pathology everywhere. And this is something we definitely learned from looking at hundreds and hundreds of ALS patient samples, right? So the ALS OX patients, they really don't have a lot of TDP-43 pathology. They have very little in the cortex. And when we see that OX signature in the core, they have very little there too. However, most of the patients in the spinal cord had elevation of both TDP-43 pathology and inflammation. So a large fraction of patients had TDP-43 pathology there. And then when you look separately in the cortex, an additional set of patients had a lot of TDP-43 pathology there. So it's not mutually exclusive, but there definitely was differences in the two systems in terms of how dense that TDP-43 pathology was in those two tissues. Right?
Thank you. We are down to our last question here. So thank you, everybody, for all the great questions you've asked. And thank you, Dr. Hammell, for sticking with us through so many really good questions as well, for such really detailed answers, clear answers. So the last question is, do you see differences in progression that correlate with the phase model? In other words, do you see inflammation mostly in patients who have progressed a lot versus mitochondria and recent symptom onset patients? So I think this also has to do with onset, but if you can untangle, yeah.
So that's exactly the one that we're trying to go for right now, right? So for example, the stage model predicts that the glia patients, the ones with systemic inflammation, should have been the ones that lasted longest, that had the longest time between when they were diagnosed and when those patients passed away, right? That actually turns out not to be the case. So there were a number of patients for whom they had highly elevated inflammatory pathway activation much earlier in the disease process, right? And so there has to be something that drives inflammatory pathway activation in some patients that isn't exclusively about stage, or at the very least isn't exclusively about time. Because some of those patients, we're talking, 10 to 18 months between when they were diagnosed and when those poor patients passed away, right? So I do think that there is something that gets inflammatory pathways active in some patients much earlier, and that is definitely associated with a very fast progression rate, right? And so trying to untangle that is the hard part, I think, right?
Thank you. We did miss one last question, actually. So we got one more for you. Natalia's got it for you.
Yeah, there is a question about your opinion about Prime C and about its premise or mechanisms, and what do you think about it? And also potentially if you have any ideas about the subtypes as well linked to Prime C.
So I don't think I know very much about Prime C. Will you tell me a little bit more about this one? There's things that I am ignorant of, and I might just be misunderstanding what you're asking me.
Prime C is a clinical therapy candidate right now that actually has had some positive results based on the last readout that they had. That is basically addressing neuroinflammation. But
is this a Healy platform trial C?
yeah, it is.
Yeah. Oh, okay. So this is presumably either the Denali or the AbbVie one or something like that, right? Yeah. So I'm excited if anything has any significant signals in the data. I think that would be fantastic, and I'd be really excited about it. I would love to either hear from those researchers or to get access to the data to see if we can see that that there was a really good signal for inflammatory pathway activation, especially in the patients who really responded strongly to that particular inhibitor. I think that is potentially a really exciting avenue. And I think we'll just have to wait and see for those to finish. I should tell you, this is the one that the disclosure, I'm on the scientific advisory board. There is going to be one for an inhibitor that targets transposons directly, and that's going to be coming up in part of one of those as well. And so I think if we look at each of those in turn and really ask, whether or not we can figure out who's responding and why, I think that's going to be really exciting.
Yeah, Prime C is specifically targeting, I think, the regulation of microRNAs. And I see a little bit more about it, but and yes, it did get cleared to go to phase 3. So we'll be looking forward to learning much more about it as it goes along. And I think we'll all be very interested to hear more about the therapeutic candidate that you're going to be potentially working on or working with. So.
Oh, no, I just advise them. They're the ones doing all of the work, but I'm just excited for anybody to have good results, right? I think we all are.
Yeah. Thank you so much. And thank you for all the time this evening, and thank you to the rest of the community. All of you have asked some great questions who've been a part of it. As always, this is half of the conversation is what you bring every week or every other week when we meet. So thank you again. And McFinn, I'll have you close us out.
All right, Molly, we appreciate more. No, how much we appreciate you. That to me is the what they call the juice that keeps you moving, is that we are all looking forward to you being healthy, for you to take care of your family, for you to do all those simple things that make you come to work and feel like, okay, I'm taking care of everything else. I'm ready for my ALS patients, and let's go. So, my dear, you're awesome, and we all believe it. And we're all hoping that you are going to help get the brass ring. So
somebody gets there for sure. And thank you so much for inviting me. I really enjoyed this conversation. I really enjoyed getting to talk to everyone.
Well, if this is your extended family, and we all want to say a wonderful evening to you. Thank you so much.